Hello all. In this session we discuss about the diuretics. Diuretics increase the elimination of electrolytes and water from the kidney by interfering with reabsorption processes at different segments. To understand the mechanism of action of diuretic, you must recall the renal physiology. So we can start with a brief outline about the renal physiology. So here you can see that there are different segments of renal tubule, the glomerulus, proximal tubular cell, descending limb of loop of Hanle, ascending limb of loop of Hanle, distal convoluted tubule, and collecting duct. Plasma which is entered through glomerulus and filtered various electrolytes. And most of the electrolytes which are filtered through the glomerulus, they are reabsorbed back by using various transport mechanism at different segment. And at the end of the day, 1.5 liter of urine is excreted out within a 24 hour. From proximal tubular cell, most of the sodium bicarbonate is reabsorbed. Almost 85% of sodium bicarbonate is reabsorbed back from the proximal tubular cell. Along with that, 40% of sodium chloride is reabsorbed, glucose, amino acid, and to maintain the osmolality, some amount of water is also passively reabsorbed from the proximal tubular cell. Here, by using specific transport pump, uric acid and creatinine, they are secreted in a tubular fluid to excreted out from the body. Here you can understand how bicarbonate ion is reabsorbed from the proximal tubular cell. As such, bicarbonate ion cannot directly cross the membrane. For that, it must require a carbonic anhydrase enzyme. So the bicarbonate ion which are present in a tubular fluid combine with the H plus ion and to form H2CO3. That H2CO3 is further metabolized with the help of carbonic anhydrase enzyme and to form H2O and CO2. That H2 and CO2 easily cross the membrane. Inside the cell, they again, with the help of carbonic anhydrase enzyme, form a bicarbonate ion and a H plus ion. That bicarbonate ion, by using a specific transport pump, cross the membrane. So this is the way bicarbonate ion is reabsorbed with the help of carbonic anhydrase enzyme. Then the thin as descending limb of loop of Henle, this is not allowed to reabsorb any electrolyte. Only water is reabsorbed from this segment, as well as from the loop of Henle, only water is reabsorbed from this side. Thick ascending limb of loop of Henle, where a specific transport pump is present, that is sodium potassium 2 chloride transport pump is present, through which sodium, potassium and chloride is reabsorbed from the tubular fluid. When this ion, sodium and potassium, they are reabsorbed in a tissue from basolateral membrane in this cell, there is again potassium ion is also diffused inside a cell by using sodium potassium ATPase pump. So inside the cell there is an increased potassium level that can be maintained by opening of potassium channel on another side, and that potassium ion is diffused back to a tubular fluid. So in a tubular fluid, increased positivity, that increased positivity can further enhance reabsorption of calcium and magnesium ion. That reabsorption of calcium and magnesium, they are not using any specific transport pump, but that is through paracellular spaces. Then at distal convoluted tubule, sodium and chloride ion is reabsorbed by using sodium chloride co-transport pump. As well, calcium ion is also reabsorbed from the distal convoluted tubule in presence of parathyroid hormone. And The last part, that is collecting duct, collecting tubule. The collecting tubule has a two type of cell. The principal cell which reabsorbed sodium ion and it can secrete a potassium ion, while the intercalated cell, they can reabsorb, they can secrete the hydrogen ion inside a tubular fluid. The cells of reabsorption of sodium and potassium, these cells are totally different than the co-transport pump which are present at other site. These are the specific channels for a sodium ion and a specific channel for a potassium ion, and their reabsorption and secretion is basically influenced by the presence of aldosterone. And at a later part of tubule, there is a effect of antidiuretic hormone. The function of this antidiuretic hormone is to synthesize more and more water channel at a collecting tubule. If more and more water channels were there, more water is reabsorbed and ultimately 1.5 liter of urine is excreted out. So both the action, the absorption of sodium and secretion of potassium is influenced by the aldosterone, and the reabsorption of water is influenced by the antidiuretic hormone. So this is a brief outline about the renal physiology. We have not discussed in detail about the major aspects, but where the diuretics are basically act, that we have covered in a renal physiology. To understand the mechanism of action of diuretic drugs, you must understand the what is a free water clearance. It is a volume of urine excreted per unit time is excess of that required to excrete the contained solute. We can say it is positive when the dilute urine is passed, and that is specifically in absence of antidiuretic hormone. And we can see it as a negative free water clearance where the concentrated urine is passed, and that is in presence of antidiuretic hormone. Both positive and negative free water clearance depends on the production of corticomedullary osmotic gradient. That means those diuretic which specifically act at corticomedullary level, they having abolishing effect on a positive as well as a negative free water clearance. Now move towards the diuretic parts. There are different class of diuretics were there. First is high ceiling diuretic. They are also known as a loop diuretic like furosemide, toracemide, bumetanide. Second one is thiazide diuretics, hydrochlorothiazide, chlorothiazide, zipamide or indapamide. That is a potassium sparing diuretic, amiloride or trimetrin, aldosterone antagonist like spironolactone, osmotic diuretics like mannitol and glycerol, carbonic anhydrase inhibitors like acetazolamide, and ADH antagonist, antidiuretic hormone antagonist like lithium. Renal physiology. In the next session, we discussed individual group of diuretics. Thank you.